Okay, let's unpack this. Welcome to the deep dive, where we really try to cut through the noise of that dense foundational science.
The kind of stuff that forms the absolute bedrock of medicine. Exactly. And today we are doing a deep dive into the engine room, cell physiology and neurophysiology. The idea here is to move beyond just, raw memorization. Right? We're aiming for real conceptual clarity. For anyone tackling high-stakes exams, or frankly, just anyone who needs a solid framework for how the human machine works, this is where it all begins.
It really is. The goal is to connect these core mechanisms, the transport, the electrical signals, directly to what you see in the clinic.
So let's start at the absolute beginning, the cell membrane.
It's so much more than just a wall, isn't it? It's this incredibly sophisticated barrier, a gatekeeper.
Well, fundamentally, it's a phospholipid bilayer. You have those hydrophilic heads, the water-loving parts.
It's facing out and in.
Yeah, facing the watery exterior and interior. And then you have the hydrophobic tails, and they form this tight, oily, water-repelling core right in the middle. And that structure that really dictates everything that follows.
And that's where the big permeability rules come from. So what gets through easily?
Anything that is lipid-soluble. So think small, uncharged hydrophobic things.
Like gases, O2, CO2.
Gases, steroid hormones. They basically just dissolve right through that oily core. It's simple diffusion, no special machinery needed.
But then you have the other stuff, anything charged or bulky, like an ion Na plus K plus R, even glucose. They run into a problem.
The big problem. They need specific machinery. We're talking about water-filled channels or pores or these dedicated carrier proteins to sort of ferry them across.
Because they just can't deal with that oily inner layer.
Exactly. Now, cells don't exist in a vacuum. They stick together to form tissues, and how they stick together is critical.
Let's talk about those attachments, starting with the tight junctions, the zonula occludens.
These are so important for creating compartments and maintaining, really steep gradients. They're like a sealant between cells.
But they're not all the same, are they? You mentioned they operate on a spectrum.
Right, a spectrum. So, for example, in the renal distal tubule, they are incredibly tight.
Which you'd need for fine-tuning everything at the end.
Precisely, for final control over ion and water reabsorption. But then you look at the proximal tubule, or the gallbladder, and they are comparatively leaky.
And that leakiness lets a lot of fluid move quickly.
It allows for bulk movement, faster absorption. It's a physiological trade-off, really. High permeability means lower resistance.
And on the flip side, you have gap junctions.
Oh, yeah.
The communicators.
Yeah, these are essentially just holes bored between adjacent cells. They permit electrical coupling.
Super important in the heart.
Vital. In myocardial cells, it lets the current just flow directly from one cell to the next, which is how you get a synchronized contraction.
Okay, so speaking of movement, let's nail down transport. Simple diffusion is our baseline. It's passive, downhill, no energy. But the second you involve protein,
the rules completely change. You enter the world of carrier-mediated transport.
What's fascinating here is that all these carrier systems, facilitated diffusion, primary active, secondary active, they all share three features that simple diffusion just doesn't have.
Right. Three defining kinetic characteristics. Let's hit them. Number one, stereospecificity. This just means the carrier protein is picky. It's designed to recognize a specific 3D shape. So the glucose carrier, for example, it'll grab D-glucose, which is what our body uses, but it will completely ignore the L-isomer.
It's a lock and key thing.
A very tailored lock and key mechanism, yeah. Okay, number two, and this is huge for kinetics, saturation. I like to think of it like toll booths on a highway. If there are only a few cars, traffic flows freely. But if you get a ton of traffic, the rate at which cars can get through is limited by the number of toll booths.
Once every carrier is busy, that's it. You've hit your max.
You've hit your transport maximum, or T. It doesn't matter how much more solute you add, the speed just won't increase. And that leads to the third feature, competition.
Right.
Right. If you have two molecules that look similar, like glucose and galactose, and they use the same carrier,
they're going to compete for those limited toll booths.
Exactly. And one will slow down the transport of the other.
Okay, so let's look at the engine that drives so much of this. Primary active transport. This is the only one that burns ATP directly to push things against their gradient.
And the undisputed champion here is the Na plus EA K plus ATPase, the sodium-potassium pump. It's the central machine of the cell.
It maintains that crazy gradient low sodium inside, high potassium inside.
It does. It typically pumps 3 sodium ions out for every 2 potassium ions it brings in. And that costs 1 molecule of ATP per cycle.
So beyond just setting ion levels, why is this pump so clinically important? What happens if it fails?
Oh, it's absolutely vital. I mean, it sets the membrane potential for every excitable cell, it maintains cell volume, and critically, that steep sodium gradient it creates. That's the energy source for almost all secondary transport.
So if it fails, the cell swells up and loses function.
Rapidly, and we see this clinically. Drugs like digitalis or oabane, they inhibit this pump to actually help the heart. It's a way to increase intracellular calcium and strengthen contractions.
Another huge one is the proton pump, the H plus K plus AT pace.
That one's in the stomach, in the gastroparietal cells.
Yeah.
Its whole job is to pump protons into the stomach to make it acidic for digestion.
And that's the target for drugs like omeprazole.
Exactly. Proton pump inhibitors. They shut that pump down to reduce stomach acid.
So that sodium gradient you mentioned, that takes us right into secondary active transport. This whole mechanism is just brilliant because it recycles energy.
It is. It's coupled transport. You have sodium, which is dying to get into the cell, moving downhill with its gradient. And the energy released from that is harnessed to pull something else uphill against its gradient.
And we split this into two types.
First, you have cotransport or symport. That's where both solutes move in the same direction.
The classic example being the Na plus cos glucose cotransporter.
The best example, yeah. In the gut and the kidney, it's how you make sure you pull every last bit of glucose back into the body.
And the second type is countertransport, or antiport.
Where the suits move in opposite directions. So the Na plus NECAC 2 plus exchanger, sodium rushes in and that drives calcium uphill and out of the cell.
Which is super important for keeping calcium levels low in heart muscle.
It all connects. What's fascinating here is how all this potential energy stored in moving ions then gets translated into these fast, all-or-nothing electrical signals, which of course brings us to the action potential.
Let's zoom in on the ventricular muscle cells, the non-pacemakers. They have that stable resting potential around nadish 90 millivolts, which is really close to potassium's equilibrium potential. So what kicks off that massive spike in phase 0?
That is the signature of the fast response. It's a huge transient opening of voltage-gated sodium channels. You get this massive inward rush of sodium current, and that's what generates the upstroke.
And then we get that long, flat part, phase 2, the plateau. Why is that plateau so important?
It's critical for the heart. It prolongs the refractory period, which is what prevents tetany, a sustained contraction. You need to give the ventricles time to fill with blood between beats.
And what's holding it flat?
It's this beautiful dynamic balance.
Yeah.
You have an inward current of calcium that is pretty much perfectly balanced by an outward current of potassium. They just cancel each other out for a while.
And then phase 3, the repolarization, the big drop.
That's when the potassium current finally wins. The calcium channel shut down and potassium conductance shoots way up. That big outward K plus current is what brings the membrane potential racing back down to negative 90.
Okay, so we've got the spike. How do we get that signal across the gap to make a muscle actually move? Let's walk through the neuromuscular junction.
So the story starts when the action potential from the nerve hits the presynaptic terminal. That depolarization triggers voltage-gated calcium channels to open.
And the calcium rushing in is the trigger.
It is the immediate trigger. That influx of calcium causes the synaptic vesicles, which are full of acetylcholine, or AAT, to fuse with the membrane and release it into the cleft.
And that HER travels across and finds its target on the muscle side.
Yes, it binds to nicotinic receptors on the muscle end plate. And these are really interesting channels because they're permeable to both sodium and potassium.
So when they open, sodium rushes in, potassium trickles out, and the net effect is a depolarization.
Right, that's your end plate potential. It moves the potential to a value somewhere halfway between the equilibrium for sodium and potassium.
We have to touch on myasthenia gravis here. It's such a perfect clinical example.
It really is. In myasthenia, you have an autoimmune attack that destroys those AT receptors. So the muscle just isn't getting the signal properly, which leads to weakness.
And the treatment is to keep the HEO that is released around for longer.
Exactly. You use acetylcholinesterase inhibitors like neostigmine. You block the enzyme that breaks down HE8, so it gives more time to find one of the few remaining receptors.
Now let's pull back to the CNS. A single neuron is getting hit with hundreds of signals at once. Synaptic integration.
And those signals really fall into two camps. You have your excitatory postsynaptic potentials, or EPSPs.
The go signals.
The go signals. They depolarize the cell, bringing it closer to its firing threshold, usually by opening those channels permeable to both Na plus and K plus ane.
And then you have the inhibitors, the IPSPs, the stop signals.
Precisely. inhibitory postsynaptic potentials. They hyperpolarize the cell, pushing it further away from the threshold,
How do they do that?
typically by opening chloride channels. The equilibrium potential for chloride is way down at like medic at 90 milliVs, so if you open those channels, negative chloride ions rush in, making the cell more negative.
Okay, let's shift to the systems level. The autonomic nervous system, the ANS.
This architecture is just essential for pharmacology. We basically divide the sympathetic and parasympathetic based on fiber length and their receptors.
The sympathetic system,
Yeah.
that's the fight or flight. It generally has short postganglionic fibers.
Right. The preganglionic releases AQT onto a nicotinic receptor in the ganglion. Then the short postganglionic fiber releases norepinephrine onto alpha or beta receptors on the organ.
But there's that one massive exception.
The adrenal medulla is a classic exam question. Its chromaffin cells are basically modified postganglionic neurons that get hit directly by the preganglionic fibers.
And then they dump epi and norepi straight into the blood.
Right, as hormones,
Mm.
for a big body-wide response.
And the parasympathetic, the rest and digest, is just the inverse.
Pretty much. Long preganglionic fibers releasing HE onto nicotinic receptors, then very short postganglionic fibers releasing HE onto muscarinic receptors right on the target organ.
And knowing those receptors is everything. Let's talk about the beta-2 receptor problem.
This is a perfect example. So beta-2 receptors, you find them in the lungs and they cause bronchodilation. That's why albuterol, a BU2 agonist, is an asthma treatment. It opens the airways.
But propranolol, a non-selective blocker.
It blocks beta-1 in the heart, but it also blocks beta-2 in the lungs. So if you give it to a patient with asthma, you're blocking their ability to keep their airways open. It can cause dangerous constriction, which is why it's contraindicated.
It's such a clear link from cell receptor to clinical decision. Okay, how does the body first even get this information? Sensory transduction.
So the job of any sensory receptor is to turn a physical stimulus, pressure, heat, whatever, into an electrical signal.
A graded signal.
A graded signal, exactly. It's called a receptor potential or a generator potential. It's not all or nothing yet. It's a measured response to how strong the stimulus is.
And only if that graded response is strong enough does it get converted into an action potential.
Right, and we classify these receptors by how they adapt. You have slowly adapting, or tonic, receptors. They're designed to detect continuous stimuli. They just keep on firing.
Like muscle spindles, telling you how stretched your muscle is.
Muscle spindles, pressure sensors, they track a steady state. In contrast, you have rapidly adapting, or phasic, receptors. They only care about change.
The onset and offset.
Exactly. They fire like crazy when a stimulus starts, and then again when it stops, but they go silent during a sustained input.
Think about putting on your clothes. You feel them at first, then the sensation just fades away.
So what does this all mean when you put it together to create movement? Let's hit those two classic spinal cord reflexes.
First, the stretch reflex, the knee jerk. It's so fast because it's monosynaptic.
The simplest possible circuit.
It is. The iafrin fiber from the muscle spindle detects the stretch, and it synapses directly onto the alpha motor neuron that goes right back to the same muscle, telling it to contract. It's for posture.
Then there's the protective one, the Golgi tendon reflex.
The inverse myotatic reflex, yeah. This kicks in when muscle tension gets too high. It's sensed by iba afferents in the tendon.
And this one is slower. Why? Because it's dysynaptic. The iba afferent talks to an inhibitory interneuron in the spinal cord, and that interneuron then inhibits the alpha motor neuron, causing the muscle to relax. It's a safety brake to prevent you from tearing a tendon.
That is just a perfect illustration of the whole hierarchy, from an ion channel all the way up to a dysynaptic protective circuit.
If we connect this to the bigger picture, every single thing we talked about, every action potential, every synapse, it all depends on the cell's ability to maintain those gradients. If that NAK pump fails, the whole system collapses.
It all comes back to that pump. Okay, let's wrap up with a quick review exercise. This will take us right back to the absolute basics from Chapter 1 on osmotic pressure.
All right, so here's the scenario for you listening. Imagine you have two solutions, and they're separated by a semi-permeable membrane. We're going to assume full dissociation.
Okay, lay it on us.
Solution A is 1 millimolar sodium chloride, NaCl. Solution B is 1 millimolar calcium chloride, KCl2. So the question for you is, assuming they both completely, fall apart in the water, which of these two solutions would have the higher osmotic pressure? And more importantly, why? The answer is all about counting the particles, the osmolarity.
Right. So solution A, the NaCl, dissociates into two particles, right? Na plus NCl. So you get 2 milliomoles per liter,
Exactly. But solution B, the key Ca2, that dissociates into three particles. You get one calcium ion and two chloride ions.
giving it an osmolarity of 3 milliomoles per liter.
So solution B, the calcium chloride solution, is hyperosmotic. It has the higher osmotic pressure because it has more solute particles. And understanding that simple relationship counting the active particles is the key to predicting how fluid is going to shift all over the body.
And that is the cellular and neural blueprint, a deep dive that gives you that clear framework for everything else in physiology that follows.